And we're back, ladies and gentlemen, talking about cell division and reproduction, part 1. Cell differentiation, the creation from one to a whole bunch of different cells. So it's taking one generic cell and developing into more specific cells. Now this can be triggered by your body, by the cell itself, or by the environment around you. It allows one single zygote. Now if you remember, zygote is a fertilized egg that develops into a multicellular adult organism that can contain hundreds of different types of cells. Again, we are taking one type and forming them into several different types of cells. So one type of cell, typically known as a stem cell, can undergo mitosis, which is cell division, and become two stem cells, or it can actually become two different types of cells, depending on the gene expression. So remember, the genes are what make you, you. So if this cell expresses the neuron-specific gene, it will create a neuron. But if this cell expresses an epithelial gene, it will become an epithelial cell. So remember, gene expression actually leads to specific types of cells. And once it has chosen which gene expression it wants to undergo, it cannot go back. So the totipotent stem cell has unlimited capabilities. It can turn into any cell it wants. It is basically our embryo. Now this totipotent stem cell can reproduce and create more totipotent stem cells, or it can go next on the list and go down to a pluripotent stem cell. These pluripotent stem cells cannot go back up to a totipotent because it's already had modifications done into the DNA. Now these pluripotent stem cells still have multiple different options they can go through and choices that they can make. They can make more of themselves, or they can come down into what are known as multipotent stem cells. Now these multipotent stem cells have made a choice, a pathway to go down. If it went down this way, it would be some sort of blood cell, blood stem cell. It could be a platelet, it could be a white blood cell, it could be a red blood cell, but it's going to be in the blood cell family. Now these other cells, again, may commit to being an epithelial cell, or a type of muscle cell. But once that choice is made, that is the path that it is going to go through. Again, it cannot go back up because the choice that is made has caused different genes in the DNA to become active and cause differences in their DNA. So when we look at differentiation, we see cells that can connect the body parts, cells that cover the lining, and other cells that can help with organs and different body parts. We need all sorts of different types. Here again, we have a stem cell that makes a path choice. Once it has decided to go down this path, it will always be a bone cell of some sort. Again, if it chooses this path, the chondrogenesis, it will create cartilage, or a chondrocyte. Again, once it is on a certain path, it cannot revert back to a stem cell state. Again, all sorts of different types that we need of cells. We have the blood cells, we have the skin cells, bone cells, epithelial cells, cardio cells, skeletal cells, smooth muscle cells, and neurons. All types that we need. So, how do we actually get these? What is mitosis? It is the division of the nucleus. It is the exact copying of the DNA in a cell. And it creates two genetically identical cells. Now, why do we need this? We need it for growth, we need it to add new tissue, replacement of dead cells, and we need it for repair. Now, mitosis rate can change as we age. As infants, we tend to undergo mitosis much quicker, therefore grow much quicker. As we get older, our mitosis or cell division slows down. Now, this might affect the rate of healing. Injuries heal faster with faster mitosis rate, so as it divides quicker, the injury will heal. But in older people with the slower mitosis rate, healing takes a lot longer than it does if you are young. Why do people die of old age? Cells undergo mitosis only a certain number of times. Once it's up, it's up. Once they divide 6 times, it's up. No more can divide after that. So the tissues, once they've finished their mitosis, die and are not replaced. There are different states of DNA when we're talking about the cell. We have the chromatin, which is DNA that is uncoiled and not distinct. It typically looks like spaghetti, and it's normally found in our nucleus. Then we have the chromosomes. The chromosomes are DNA that is coiled into individual segments, and it looks like an X. So this is the chromosome right here. Now the chromosome is made up of 2 sister chromatids. One chromatid is going to be one side, so one upper and one lower. And then the second sister chromatid will be the second upper and lower. Chromosomes are only going to be found as a part of mitosis. Again, the DNA is coiled around a histone, making a nucleosome. 6 nucleosomes come together to make a flower or ring pattern, and that's going to be our chromatin. That is what's going to be in our normal nucleus. Now, when the chromatin starts looping around and using protein scaffolding to loop and coil, it creates a chromosome. The center of the chromosome that holds together the sister chromatids is called the centromere. Then we have the 2 sister chromatids, again, one lower part and one upper part. That's one sister chromatid. And then another sister chromatid, one upper, one lower. They are exactly identical. During the cell cycle, most of the time our cells are in interphase. That's what takes up most of the time of our cells. And specifically, most of the time in G1. Our cells, once they're told that they need to divide, will then go into S phase, which is DNA synthesis, into the G2 phase, and then into mitosis and cytokinesis to produce 2 new cells. Again, G1 is going to be growth and normal metabolic role. So it's normal functions. It's just sitting there doing its normal thing, growing, doing its normal thing. It gets a signal that says, hey, you need to go through mitosis. You need to divide. So it goes into the S phase, which is DNA replication. So DNA is replicated, the organelles are replicated, and then it goes into G2, which is growth 2 and preparation for mitosis. So it gets everything prepared to go through mitosis. Mitosis has 4 parts to it: prophase, metaphase, anaphase, telophase. Hemat. And then directly after it, that's not shown here, is known as cytokinesis. Cyto meaning cell, kinesis is meaning to split. So again, interphase is our normal start, everyday cell. It's just chilling. Then it gets a signal, and it starts going into prophase. We have shown here prophase early and prophase late. Then we have metaphase. The chromosomes are lining up in the middle. Then we have anaphase. The chromosomes are being pulled apart. And then we have telophase and cytokinesis, 2 new cells being formed. Again, mitosis is all about the genes, the DNA being copied and separated. Only when we hit cytokinesis do we care about the organelles or any of the other parts. But remember, mitosis is talking about DNA. Cytokinesis is everything else splitting. Here are some images. Interphase is a normal cell. Prophase, we start seeing some of those chromosomes or chromatin being formed. Late prophase, we actually do see the chromosomes. We see the distinct chromosomes. Metaphase, they're lined up in the middle. Anaphase, they're being pulled apart. Telophase and cytokinesis. The cell begins to pinch with a cleavage furrow. Organelles are separated, and the nucleus is being rebuilt to form 2 new cells. Again, we see the stages again. Interphase, not part of mitosis. Majority of the cell's life, it's growth, metabolism, DNA replication. G1 is first growth, S is synthesis, and chromosomes are copied, and G2. Looking a little bit further in, what does the cell does not undergo mitosis? This is what it looks like when it's not in mitosis. If the cell receives the signal, DNA is replicated during the S phase, organelles are added and created during the S phase, and you can tell that it's in interphase because there are no discrete or distinct chromosomes visible. Prophase. Prophase, you'll see both early and late, but we have chromosomes becoming visible. Chromosomes becoming visible. The nuclear envelope or nuclear membrane breaks down. Centrioles move to opposite ends of the cell, and spindle fibers form. The easiest way to remember this is prophase is CNCS. Chromosomes appear, nuclear envelope disappears, centrioles move to opposite ends, and spindle fibers appear. CNCS. Metaphase in the middle. Metaphase in the middle. Chromatids line up in the middle. The spindle fibers attach to the chromatids in the centromeres, and they help align them on the equator, or the center or middle of the cell. Anaphase apart. Anaphase apart. Sister chromatids are pulled apart. Spindle fibers attach to the chromatids and are shortened, and therefore pulling them apart. And the other spindle fibers start lengthening the cell and pushing the cell membrane apart. So you see it here becoming longer and elongated as the chromatids are pulled apart. Telophase and cytokinesis. Telophase is the opposite of prophase. So chromosomes unpack, nuclear envelope reforms, centrioles divide and go into their opposite, or go with their cells. Spindle fibers break down, disappear. The opposite of prophase. Cytokinesis is the actual splitting of the cell. All the organelles in the cytoplasm are split. Now, in animal cells, we have a cleavage furrow that forms. That is going to be that little dent. It's known as a cleavage furrow. In plants, we see a cell plate form in between. Review. Prophase, metaphase, anaphase, telophase. Prophase, CNCS. Metaphase is middle. Anaphase apart. Telophase is the opposite of prophase. Cell regulation. Again, most of the time your cell is in G1. It's doing its normal thing. Once it gets that signal that it needs to go through mitosis, it will go through the G1 checkpoint, which checks for cell size, nutrient, growth factors, and it checks damage for the DNA. If that's all good, it can go into the S phase, which is DNA synthesis, which copies the DNA, copies and makes more of the organelles, and comes into the G2 phase. At the end of the G2 phase, we have our second checkpoint, which is called the G2 checkpoint. It checks for the cell size, and it makes sure that DNA replication has occurred and that it is correct. Our third and final checkpoint is going to be the M checkpoint, which it checks to make sure that the spindle fibers are attached to the center of those chromosomes, so that when the chromatids are pulled apart, the spindle fiber is actually attached and can pull it apart. Well, the cycle continues, but this is the end for now. See you later.